The overall goal of the following experiment is to localize proteins in electron micrographs using correlated fluorescence and electron microscopy. Here, this method, known as Nano-fEM, is used to localize proteins in C. elegans. First, a transgenic worm expressing histone TDEOS is embedded in plastic and sectioned. Each section is imaged by both photoactivated localization microscopy, or PALM, and scanning electron microscopy. The two micrographs for each section are aligned using fiducial markers using Photoshop. The correlative image shows the localization of histone molecules in the nucleus.
So the main advantage to the technique that we are going to describe today is that instead of using immuno-EM, we are using genetically encoded fluors so that every protein is associated with a fluorescent protein. When dealing with immuno-EM, many of the antibodies can't act with plastics. It's just not compatible. And if it is, the antibodies are not able to penetrate deep into the section where the antigens are. By using fluorescent proteins, every protein is tagged with a fluor. So today, Shigeki Watanabe will be demonstrating this technique.
The following procedure will be demonstrated using transgenic nematodes that express histones tagged with the fluorescent protein TDEOS.
For this demonstration, we will use bacteria as a cryoprotectant, but you can also use 20% BSA for freezing C. elegans.
1 hour before freeze substitution, fill the automated freeze substitution or AFS device with liquid nitrogen. Also in advance, prepare the needed fixatives according to the instructions in the accompanying document. Store the fixatives in 1 ml aliquots in cryovials and place them in liquid nitrogen until they are used.